Hello friends, welcome to my video. In this video, we are going to see about bacterial nutritional requirements, sources of nutrition for bacteria and classification of bacteria based on nutritional requirements. We usually cultivate and study bacteria under laboratory conditions. One exception is ecological studies. For cultivating bacteria, a knowledge about bacterial nutritional requirement is very important. Bacterial nutritional requirements vary widely. So, for the successful cultivation of bacteria and awareness of their nutritional requirements and physical conditions favoring growth such as temperature, pH and gaseous environment is required. Bacteria have their own nutritional requirements for growth and normal functioning. Bacteria require a source of energy, a source of electrons, a source of carbon, and also require nitrogen, oxygen, sulfur, phosphorus, metal ions, vitamins, and water. Source of energy. Sunlight and chemical compounds are the two major sources of energy. Source of electrons for their metabolism. Organic compounds and reduced inorganic compounds are the major source of electrons. Source of carbon. Organisms require carbon for synthesizing cell components. Carbon dioxide and organic compounds are the sources of carbon. All organisms require at least small amount of carbon dioxide. Source of nitrogen. Bacteria require nitrogen in some form for cell components. Some bacteria can use atmospheric nitrogen. Some bacteria can use inorganic nitrogen compounds such as nitrates, nitrites or ammonium salts, and some bacteria can derive nitrogen from organic compounds such as amino acids. Source of oxygen. Oxygen is provided in various forms such as water, component atoms of various nutrients or molecular oxygen. Source of sulfur. Sulfur is needed for synthesis of certain amino acids. Some bacteria can use organic sulfur compounds. Some can use inorganic sulfur compounds and some can use elemental sulfur. Source of phosphorus. Phosphorus is an essential component of nucleotides, nucleic acids, phospholipids, etc. Phosphorus usually supplied in the form of phosphate. Metal ions. Bacteria require metal ions for normal growth. Bacteria needed some metal ions at very low concentrations. So, these are termed as trace elements. These trace elements present in other components of culture media in sufficient amounts to support bacterial growth. So, it is not necessary to add these trace elements to the media. Some of these metal ions are known to be cofactors for various enzymes. Source of vitamins. Bacteria require vitamins and vitamin-like compounds. These function either as coenzymes for several enzymes or as the building blocks for coenzymes. Some bacteria are capable of synthesizing their entire requirements of vitamin, and some bacteria cannot synthesize it and will not grow unless the required vitamins are supplied preformed to them in the medium. Source of water. Bacteria require water. Water is the vehicle of entry of all nutrients into the cell and for the elimination of waste products. And the source of water is moisture. Next, we are going to see some important points about sodium ion. Most bacteria do not require sodium ion. Exceptions are certain archaea and cyanobacteria. Some archaea cannot grow with less than 12 to 15% sodium chloride. They require the high level of sodium chloride for maintenance of the integrity of their cell wall and for the stability and activity of certain of their enzymes. Up to this, we learnt what are all the nutritional requirements of bacteria and sources of nutrition for bacteria. Next, we are going to see about classification of bacteria based on nutritional requirements. Classification of bacteria based on source of energy. As I told you, sunlight and chemical compounds are the two major sources of energy. Bacteria which derive their energy from sunlight are called phototrophs. Bacteria which derive their energy from chemical reactions are called heterotrophs. Classification of bacteria based on sources of electrons. We know that organic compounds and reduced inorganic compounds are the major sources of electrons. Some bacteria can use organic compounds as electron donors and are called organotrophs. Organic compounds, organotrophs. Some bacteria can use reduced inorganic compounds as electron donor and are called lithotrophs. Classification of bacteria based on source of carbon. Carbon dioxide and organic compounds are the sources of carbon. Bacteria which can use carbon dioxide as their major source of carbon are called autotrophs. Bacteria which can use organic compounds as their carbon source are called heterotrophs. Next, we are going to see about phototrophs. Some phototrophic bacteria use inorganic compounds as their source of electrons and are called photolithotrophs. Some phototrophic bacteria use organic compounds as their source of electrons and are called photoorganotrophs. Some phototrophic bacteria under certain circumstances can grow as a hemotroph. Next, we are going to see about heterotrophs. Some hemotrophic bacteria can use inorganic compounds as their source of electrons and are termed hemolithotrophs. Some hemotrophic bacteria can use organic compounds as electron donors and are called hemoorganotrophs. Some bacteria can grow as either hemolithotrophs or hemoorganotrophs. Next, we are going to see about autotrophs. Autotrophic bacteria exhibit the simplest nutritional requirements. Autotrophs are able to utilize atmospheric carbon dioxide and nitrogen. Autotrophs can transform a mixture of inorganic compounds into the carbohydrates, proteins, nucleic acid, lipids, vitamins, etc. Autotrophs plays an important role in cycling of elements through biological system. And a very important point to be noted about autotrophs is that autotrophs are capable of independent existence in water and soil and are of no medical importance. Autotrophs plays an important role in agriculture and the maintenance of soil fertility. Some autotrophs are facultative autotrophs. They can either live as autotrophs, deriving their carbon from carbon dioxide, or they live as heterotrophs, deriving their carbon from organic compounds. Some autotrophic bacteria derive their energy from sunlight and are called photoautotrophs. Photoautotrophs have chlorophyll pigment to capture sunlight. Some autotrophic bacteria derive their energy from chemical reactions and are called hemoautotrophs. Photoautotrophic bacteria played an important role in the evolution of an oxygen-rich atmosphere on Earth. Heterotrophs. Heterotrophic bacteria have been studied more extensively than the autotrophs because heterotrophs can cause diseases of human beings, other animals and plants. Cultivation of heterotrophs. Depending on the species, the nutritional requirements may be relatively simple or complex. Heterotrophic bacteria are unable to grow with carbon dioxide as the sole source of carbon. The nutritional requirements of heterotrophs vary widely. Some may require only a single organic substance such as glucose, while others may need a large number of different compounds such as amino acids, nucleotides, lipids, carbohydrates and coenzymes. Among heterotrophs, some bacteria have elaborate requirements for specific nutrients, and such heterotrophs are designated as fastidious heterotrophs. Some heterotrophic bacteria can use light energy and are called photoheterotrophs. Some heterotrophic bacteria derive their energy from chemical reactions and are called hemoheterotrophs. Hemoheterotrophic bacteria are further classified into saprophytic bacteria and parasitic bacteria. Saprophytic bacteria, those bacteria which depend on dead and decaying matters, are called saprophytic bacteria. The saprophytic bacteria are also known as decomposers because these bacteria can convert complex organic substances into simple chemicals. Parasitic bacteria. Parasitic bacteria obtain their nutrition from the host tissues on which they grow. Some are harmless and some causes serious diseases in plants, animals and humans. Finally, we are going to see about obligate parasites. Obligate parasites have not yet been successfully cultivated on an artificial medium, and their physical and nutritional requirements are not understood. As I told you in my first video, microbiology is the field of science with immense scope. So learn it, make great discoveries and help the field to grow. Thank you friends.